Okay. Um, Anyhow, so for today's Idea Forum, what we're excited to hear about is some presentations from our pre-college students, which, I guess that goes pretty far down the line, but in this case, it's high school students who are doing Alzheimer's and related dementia research and outreach work. And we're really excited about this. One of the things that the REC cares a lot about and that the NIA ADRC program cares a lot about is trying to enhance the sort of pipeline of young investigators who are interested in Alzheimer's disease and dementia care, because convincing somebody in their third year of medical school is certainly doable, but it is probably not as high return on investment as doing it earlier on in the process. So anyways, we're really excited about this, getting to do that and to also to have them share what they've been working on. So today we'll be hearing from Bianca Chan from Kajal Parmar and Danny Topper, and we will be going in that order. So up first is going to be Bianca. So she is a high school senior at the North Carolina School of Science and Mathematics, which is right here in Durham. Go Unicorns. I think actually for Effecti, it's worth pointing out that it's oftentimes difficult to have high school students work on projects because you can't add anybody to an IRB or let them have access to PHI if they are under the age of 18. And so for that reason, when I've had high school students do work, oftentimes we're using like summary statistics from genomic studies or whatever, but it's all sort of de-identified type of information. And so one thing I'd just point out is that the School of Science and Math actually has now partnered with Duke and our whole regulatory core over in Docker. And so we still can't add people to IRBs and access PHI from high schools, with the exception of students enrolled at NC Science and Math, which is kind of cool. So good on them for doing that. Okay, so back to Bianca. So high school senior at Science and Math, and she has conducted dementia research with Joshua Kane and Michaela Brown, who are Southern California, UCLA, I believe. And I believe also did summer research internship at Hopkins, and her interests really reside in imaging-based biomarkers for Alzheimer's disease, as well as with what I think she's going to talk more about today, non-pharmacologic interventions in dementia care, and in improving quality of life for patients and caregivers as well. So today she's going to share the story behind the Neurologic Foundation that she established. And I think it's also a really nice complement to some of the work that Melissa Harris, one of our rec scholars who's now on faculty at the Durham VA, is also doing. So with that, I will stop and I'll go ahead and let Bianca get started. Last announcement. We'll try to do maybe like one question at the end of each presentation, and then try to reserve, 10-ish minutes or so for discussion with the whole group at the end. So with that, Bianca, all yours.
Hi, everyone. My name is Bianca Chan. I'm originally from Cary, but I currently attend the NC School of Science and Math. So thank you for having me talk about my nonprofit today. So I'll be sharing about how I founded this nonprofit and kind of my journey going forth. So I thought I would first share about how I was introduced to Alzheimer's and how my journey began with advocating for this disease. So I remember that in freshman year of my high school, I came across a news article with a video of a former ballerina with severe memory loss. I don't know if you guys are familiar with this news article, but personally, when I looked into the video, I was really touched by this ballerina's emotional vulnerability. So in the video, basically, the ballerina was dancing along to a song called Swan Lake. And by her hand movements, I really saw the power of music in igniting these past memories. And so afterwards, I searched for more stories of those with Alzheimer's, just like this ballerina. And I saw that the impact that this illness had on patients themselves and also their loved ones. But more importantly, I became really curious about how the brain changes with Alzheimer's disease and how that correlates with behavioral changes. So I became really interested in the scientific aspects of it. So that following summer, I applied and joined a research program called the Lumiere Research Program. So in that research program, I proposed to study the biomarkers of Alzheimer's, so the functional and structural biomarkers of Alzheimer's detected using brain scans. And I was paired up with my mentor, Dr. Joshua Kane, who is a neuroscientist from UCLA. So alongside his guidance, I used 3 types of brain scans: structural MRI, functional MRI, perfusion MRI, and then PET scans from the Alzheimer's Neuroimaging Initiative to conduct my study. So these scans were used to detect patterns of brain changes and exactly what regions underwent the most changes due to the progression of the disease. For example, I used structural MRI to find what regions had the greatest amount of atrophy or brain tissue loss, and what regions had the most amount of ventricular enlargement, which are 2 primary structural biomarkers. I then measure them alongside the rate of change in healthy elderly. And then I also use functional MRI to measure the decrease in default mode network activity and overall decrease in cerebral blood flow. And lastly, I use PET scans to track hypometabolism and the decrease in the overall brain glucose consumption in certain regions of the brain. So from this project, I was able to validate a lot of the biomarkers traditionally seen in Alzheimer's brains and gain a more thorough understanding of this disease from a biological standpoint. So while I was conducting this research, I've had the chance to personally interact with a lot of patients and caregivers who are affected by Alzheimer's disease or memory loss in some other form. So through these conversations, I learned a lot about their stories and their experiences. And a lot of people mentioned that the cost of Alzheimer's treatment and drugs are quite high, which could place a burden on families, especially those who are already economically disadvantaged. And some families also mentioned to me that since they lived in more rural parts of the state, they found it difficult to access certain support and care services to help them cope with these lifestyle changes and the memory loss of their family member. So because I saw that geographical locations and financial barriers could greatly affect these families' ability to access quality care in Alzheimer's, I really dug into the questions of how I can use what I've learned already and my current skills to help them access these services. So I decided to talk with more social workers at different local senior agencies and associations like the Dementia Alliance of North Carolina and the local chapter of the Alzheimer's Association. And I understood more about what I could do to advocate and help bridge these gaps in a way that's entirely free and in a way that's easily accessible. So ultimately, these personal stories are what motivated me to get involved in not only the research component, but also advocate within the community itself. So after talking to these social workers, the first initiative that I created was creating custom packages known as neuroboxes. So each package contained items like fidget toys, squishy toys, and also coloring books, puzzles, green tea packets, and more. And I chose to incorporate these items because when I researched on the therapeutic effects of these items, they've been found to alleviate some of the cognitive symptoms of Alzheimer's and also the behavioral symptoms like anxiety and agitation. And it has also been found to improve hand-eye coordination in some patients. So especially some of these items like green tea, which you wouldn't think traditionally would affect the brain, has been found to reduce the buildup of proteins in Alzheimer's brains. And to create these boxes and to prep for these items, me along with some other volunteers, we did a lot of fundraising through walkathons and also local bake sales. So these boxes are not only intended to be used by the patient themselves, but also with their caregivers or loved ones to help alleviate the stresses of caretaking, which I understand can be overwhelming at times. So I remember that one time I was spending time in a memory care facility, and I saw that a patient was working on a puzzle from a neurobox with his grandson, and I just realized how therapeutically powerful even these social activities can be for maintaining intergenerational connections. And one of the reasons why I chose to create this initiative, this Neurobox initiative, is that it allows these individuals to have a voice for themselves and still preserve their dignity through choosing what activities that they want to engage in. And I believe this is super important because these individuals often feel isolated because of their struggle to communicate. And since then, I've donated hundreds of these boxes to 20 different senior care facilities in North Carolina. And when I go to college and in future years, I really hope to expand this initiative to donating more boxes in other states as well. So a few months later, I then began a music entertainment program called Neuro Melodies. So through this program, I have recruited both professional musicians and also student musicians locally and both out of state to play music in long-term care facilities. And so these long-term care facilities both are located in North Carolina and also in different states around the nation. And recently, I've tried to incorporate specific audio frequencies within our music because I read that in one study by MIT, audio frequencies of around 40 hertz have been found to help break down these protein buildups in Alzheimer's brain. So this is something that's relatively new and has not been done in a lot of traditional music therapy services. So this is definitely something I wanted to explore further. So we frequently host these programs through both in-person and virtual sessions, and then we also have pre-recorded videos. And our volunteers usually play music of different lengths and different genres because we understand that each individual has different music preferences and also attention spans. And our music has been used for several Tai Chi classes and other projects at different senior care agencies around the country. And as of this year, we have reached around 2,000 Alzheimer's patients across the nation with our program. So last year, we then began partnering with trained dementia care groups, support group facilitators to start offering support gatherings for both families in both online formats and in-person formats. So on the right here are just some of the pictures of the gatherings that we have offered so far. And usually in these gatherings, patients themselves, their spouses, their children, friends, their other loved ones have joined. And these gatherings are intended to be like a stigma-free space where they can socialize and engage in activities like Zumba, puzzle solving, poetry reading, and singing. And they usually range from around 10 patients to around 20 people per session. And usually I'm the one leading these events, but I also have trained professionals with me and also some volunteers helping out with the prepping. And for me personally, it's always great to see that these individuals have a support network that can help them thrive through the challenges of the disease. And it's really fulfilling to create a platform where people can connect with other people going through similar struggles on a more personal basis. So now in my senior year through my school, I've had the special opportunity of working with a facility under the UNC Blue Ridge system called the Grace Ridge Retirement Community, which is also a continuum of care center. So there I spend around 2 days a week working alongside a geriatrician, Dr. Plyler, and the executive director, Michaela Brown, to research the effects of the Montessori method on the cognitive functions of dementia patients. So through this research, I really wanted to improve my initiatives because my initiatives are centered around that mission of Montessori. So last month, our nonprofit helped run a flu clinic by working with Dr. Plyler and Ms. Brown and some geriatric nurses under the UNC Blue Ridge system to help 50 elderly residents get vaccinated in that center. And so in that project now with the Montessori method, I'm studying with particularly with 10 residents diagnosed with dementia at the center. And at the end of my research at that facility, I hope to use my findings to further outreach the public by developing educational programs on the Montessori method and on using non-pharmacological tools in dementia care. And as I work closely along with these 10 residents, I hope to use my school's fabrication laboratory to create sensory stimulation boards, which includes hand-on activities like I know there's like a zipper activity, and then it helps these individuals improve their motor skills and their hand-eye coordination, which has a similar mission as some of the activities that I included in our neuroboxes. So as I've continued to research and engage in community work in the Alzheimer's field, I want to acknowledge some of our partners who have supported our nonprofit since its founding and helping us expand. So my partners include local networks like Dementia Alliance of North Carolina and national agencies like the Alzheimer's Association and the National Council on Aging. And I also want to thank the memory care homes and the facilities that have allowed us to host these programs in their centers. And I'm especially grateful for my mentors, Dr. Joshua Kane and Michaela Brown, and the social workers, the geriatric care professionals, and the dementia specialists who have guided me in helping me develop this nonprofit and my initiatives. So thank you for listening to me talk about my presentation, for talking about my work. Does anyone have any questions?
That is an impressive body of work.
Let me just second that. That's really impressive work and really, really great. I think it would be nice to know if I'll just ask one question and then others can ask later. What challenges you had with getting this up and going and implementing? Because I think that derails some people sometimes, the daunting task of getting the research launched.
Yeah, definitely. So when I was first starting my programs, I found it difficult to get a team together because I understand that not everyone is passionate about the same thing and not everyone has the same idea of how to help these patients. So mainly I work with both volunteers and other students in online formats. So they include people from both my school and also people from out of state. And so basically we always have monthly meetings to kind of talk about our plans. So which I found has been really helpful. So just getting that like teamwork and that making sure that communication is smooth has definitely been a struggle, especially in the beginning of founding my nonprofit. But as we've met more and as we've grown closer, I feel like that network has also grown stronger.
Wonderful. Thanks for a really fun presentation.
Thank you.
Okay, so next we're So next is Kajal. So Kajal is a sophomore, so 10th grader at Carey Academy. And she's actually working with me, so I'm not going to ask a question at the end because I get to do that to her all the time. So she's particularly interested in gaining a better understanding about the biology of neurodegenerative diseases. And because she does some work with me, we tend to also look at brain tumors as well. So her talk today is going to be on a sort of semi-systematic review that we're doing, looking at differences in Alzheimer's disease and glioblastoma as it relates to microglial function and polarity. So it's been a pleasure working with her this year, and we're looking forward to your feedback and thinking about these ideas as she sort of works towards assembling this into a review article. So,
thank you, Dr. Walsh. Let me pull up the slides. As Bianca said, thank you all for being here. Just echoing what Dr. Walsh said as well, my name is Kajal Parmar, and I'm a sophomore at Carey Academy. And today I'll be going over a microglial review in Alzheimer's disease and glioblastoma. So this work is part of the Duke Neuroepidemiology Laboratory, which specifically focuses on early life origins of disease, genetic and immunologic predisposition, as well as biological processes associated with health and diseases that have occasionally divergent evolutionary constraints. So this table is what most of our research is focused on, and it describes the inverse relationship between factors in glioblastoma and Alzheimer's disease. As I said, Dr. Walsh and I have based this project off of this table and demonstrates the risk factors which increase or decrease risk for GBM and Alzheimer's disease. And then this information on the table has been previously available, and we're looking to use this to move forward when it comes to a microglial perspective. The motivation of this study is that Alzheimer's disease is associated with elevated neuroinflammation, and glioblastoma is associated with reduced inflammation-mediated tumor immunosurveillance. The objectives are to determine whether the factors in Table 1 are associated with pro- or anti-inflammatory states in microglia. And lastly, to determine whether relationships are consistent with elevating risk factors of one disease or decreasing risk of another. And finally, the impact is to assess the plausibility of Alzheimer's disease and glioblastoma associations in Table 1 being mediated through their pro- or anti-inflammatory effects on microglia. So to get started, female sex is typically associated with lessened risk for glioblastoma and increased risk for Alzheimer's disease. We see an increased expression of M1 markers, with major histocompatibility complex 2. These M1 markers and macrophages inhibit cell proliferation and are immunosuppressive, whereas M2 markers are promoting cell proliferation and tissue repair. We also see increased phagocytosis, microglial activation, which is seen with inflammation, and increased TIM3 expression, which is a mediator of neuronal apoptosis. So when it comes to these M1 markers, they're immunosuppressive macrophages. So they can actually mediate cytotoxicity to kill cancer cells with these pro-inflammatory cytokines. But it's these cytokines that increase neurodegeneration in Alzheimer's disease. Next, when it comes to phagocytosis and activation, this can support the T cell response against cancer cells and kill these cancer cells, whereas increased phagocytosis in Alzheimer's disease can kill healthy synapses and processes of life, again, leading to further neurodegeneration. And lastly, for TIM3, this inhibits cancer progression and proliferation, whereas in Alzheimer's disease, TIM3 can contribute to amyloid beta accumulation, which is a key pathological factor in Alzheimer's disease, also increasing inflammation. Next up, we studied age, specifically over the year of 75, and that seemed to actually reduce GBM and increase Alzheimer's disease. With age, we can see natural glial senescence and depletion, oxidative stress and oxystrol production, dystrophic microglia, myelin impairment, the aggregation of asynuclein, increased M1 macrophages and MHC2, and then the decline of microglial homeostatic functions due to the reduction in TGF-B1, which is a potent chemotactic cytokine. So this senescent and depleted microglia in GBM can prevent the proliferation of pre-malignant cancer cells and reduce tumor vessel count. But in Alzheimer's disease, senescent microglia, as well as dystrophic microglia, asynuclein, myelin impairment, it's linked to tau aggregation. And tau is another major pathological factor in Alzheimer's disease. And it also increases their pro-inflammatory response. In GBM, oxidative stress and oxystrol, as well as M1 macrophages with MHC2, through increasing inflammation, they can inhibit tumor growth. Whereas in Alzheimer's disease, this oxidative stress is linked to senile amyloid beta plaques. And lastly, asynuclein, which promotes inflammatory responses, can lead to the recruitment of macrophages in tumors, helping the immune response. But in Alzheimer's disease, as I said earlier, asynuclein can lead to more inflammation, and this inflammation also comes from the M1 macrophages, reduced TGF-B1, and more oxidative stress. Next, we looked at European ancestry, and we used the knowledge of the fact that European ancestry typically decreases one's risk for GBM, but increases risk for GBM and decreases risk for Alzheimer's. There's not enough research specifically focusing on risk factors with those of European ancestry. So Dr. Walsh and I examined studies looking at factors increase in those with African heritage and used the converse relationship to examine non-Hispanic whites. Historically, we've seen that those with African heritage are more susceptible to Alzheimer's disease and less likely for glioma, and then the opposite relationship in those with European ancestry. And this slide is more relative for Europeans, and it can be inverted for those of African heritage. So we've seen that non-Hispanic whites have higher TREM2 levels, and TREM2 regulates inflammatory processes. They also have higher levels of gluconeogenesis and glycolytic signatures. They have stronger phagocytosis levels in response to fibrillar amyloid beta tangles, and an increased cytokine response to LPS, which actually causes neurodegeneration. In glioblastoma, TREM2 levels can increase cell proliferation, which causes like the rapid increase of cancer cells. Whereas in Alzheimer's disease, these TREM2 levels, in addition to phagocytosis to specifically amyloid beta, can accelerate the immune response and reverse Alzheimer's disease neuronal phenotype. Additionally, in GBM, gluconeogenesis and increased glycolysis can upregulate GLUTs, which are a specific transporter in cancer types, whereas in Alzheimer's disease, it's linked to neuroprotection, which is actually really interesting. In GBM, this increased response to LPS reduces survival rates and reduces that anti-tumor immunity needed. Whereas in Alzheimer's disease, it can reduce amyloid beta, reduce oligodendrocytes, reduce myelin injury, and even inflammation, and all these factors that would contribute to Alzheimer's disease, but this response actually reduces it. Next, we looked at varicella and herpes virus infections, and we've seen that it decreases risk for GBM, but increases risk for Alzheimer's. So this effect on microglia is that it induces the process of encephalitis, which is inflammation in the brain. It increases phagocytic activity, sustains chronic neuroinflammation and inflammatory cytokines. It increases oxygen radicals, and then it activates, again, that M1 microglial phenotype. There's currently a lot of growing research on encephalitis reducing glioma risk through anti-tumor immunity. But of course, since encephalitis is associated with inflammation, that leads to neurodegeneration when it comes to Alzheimer's disease. The increased phagocytosis and cytokines in GBM can support the T-cell response and provide benefits in the glial microenvironment, but these cytokines in Alzheimer's disease, like TNFA, and then other things such as IL-1B, other interleukins, and the M1 phenotype can lead to inflammatory responses. In GBM, the increased oxygen radicals can lead to the regulation of glioma growth, whereas in Alzheimer's disease, this radical production damages microglia and increases amyloid beta production and inflammation. And then in GBM, we see this M1 phenotype expression playing an anti-tumorigenic role. And then to add on to Alzheimer's disease, this excessive phagocytic activity can damage healthy synapses and also healthy neurons. But in addition to that, it can kill too many neurons, and then it can kill things beyond just infected cells, leading to neurodegeneration. Next, we studied type 2 diabetes, which have a decreased risk for GBM and increased risk for Alzheimer's. For type 2 diabetes on microglia, it upregulates activity markers such as IBA1, CD68, and major histocompatibility complex 2. It also upregulates pro-inflammatory cytokines and chemokines, increased oxidative stress, hypertrophic and amoeboid microglia. And then we often see microglia trapped in this state of metabolic stress. And then also the RAGE microglial pathways overexpressed. So in GBM, this increase in MHC2 can lead to anti-tumor immunity through the CD4 plus T cell lymphocytes. Whereas in Alzheimer's disease, MHC2 can increase cognitive deficits and degradation. And that's in addition to IBA1 and CD68. The increased hypertrophic microglia in GBM secretes those pro-inflammatory cytokines to actually kill tumor cells, whereas in Alzheimer's disease, these pro-inflammatory cytokines can cause significant neuronal damage. And then when it comes to the increased oxidative stress, it can create neurotoxicity, thus increasing risk for Alzheimer's disease. And lastly, we have telomere length. So much research that's already been conducted on telomere length is examining the effect of shorter telomeres. So this used studies when looking at longer telomere, or this used studies about the shorter telomeres, but we looked at it from a longer telomere perspective, which increases risk for GBM and decreases risk for Alzheimer's. First, we saw that there were increased TERT levels, which are telomerase reverse transcriptase, which is a subunit of telomerase, a reduction of pro-inflammatory cytokines, decreased TDDR, which is telomeric DNA damage response and microglial senescence, and then increased microglia proliferation. In GBM, this presence of TERT can prolong the longevity of tumor cells compared to Alzheimer's disease, where it can enable the resistance to pathological tau and prevent more neuronal damage. And in GBM, this microglial proliferation can augment the growth of GBM and increase the cycling of malignant cells. Whereas in Alzheimer's disease, we've seen that telomeric attrition increases DNA damage response, which increases these pro-inflammatory cytokines, senescence, and oligomers. So therefore, with longer telomeres, this attrition is decreasing these pro-inflammatory cytokines and thus decreasing the risk for Alzheimer's disease. So our key takeaways on this study are that many of the risk factors that are associated with elevated Alzheimer's disease risk and reduced GBM risk are known to influence microglial states. And while not universally so, the way that these factors influence microglial states generally corresponds to whether these factors lead a more inflammatory or non-inflammatory microglial state, and then typically aligns with their direction in GBM and Alzheimer's disease. And lastly, microglia may mediate the potential relationship between these factors and risk or protection against GBM and Alzheimer's disease. Here are just all of the PMIDs that we use so far for each of the risk factors. And thank you to all of the collaborators and funders for this project. And most importantly, thank you to Dr. Walsh for always supporting me and guiding me through this process. And we're very excited to see where this goes in the future.
Great. Okay, I'm not allowed to ask a question because I do that to her all the time. But if we don't have a question now, we can save them for the end. So if anyone wants to speak up or raise your hand, go for it.
I have a question.
Shefali Parmer?
Hi. So where do you see, sort of after looking at all of the different factors with Alzheimer's disease, where do you see this sort of research and sort of the future of your findings?
So in the future, we're going to look at like therapeutics targeting activated microglia in GBM and suppressing or reprogramming microglia, and then seeing these two research fields stand to interact more in the future.
That's great.
Thank you, Kajal, and we will come back with more questions. You know, the problem with a lit review that is this intensive is you kind of flood people with data, but you did a really excellent job with sort of the conclusion slide of, you know, how this sort of all kind of fits into a common picture. Also, there were a lot of, you know, $1,000 words that you had to pronounce, and I think you nailed like 99.8% of them. So it's excellent. All right. So our third speaker today is going to be Danny Topper, and I believe Sylvia Smith from UNC Pembroke, who has guided her in her research, is on to give her an introduction.
I'm here. Hi. Hello, everybody. I'm Sylvia Smith. I'm a human biologist by training. I use some bioinformatics tools to study various chronic inflammatory disorders. A couple of years ago, I started working with Danny Topper, who's a senior now in high school at Harrison High School in New York City. Excuse me, in the state of New York. Danny is one of those outstanding students. She's a summa cum laude student. and she's actually gearing up to go to college next year. Well, she will be studying biomedical engineering. Let's see. She tells me that she enjoys taking naps, but I don't suspect that she actually has a lot of time to do that based on her work and my experience in working with her. So she has been working with Dr. Barr and I on Alzheimer's disease, and she's going to tell you about it in just a second.
Thank you, Dr. Smith. Let me just share my screen. So as Dr. Smith said, my name is Danny Topper, and I'm a senior at Harrison High School in Harrison, New York. And I collaborated with both Dr. Ben Barr and Dr. Sylvia Smith from UNCP on my project. And my presentation is entitled Evaluating Gene Linkages Through Pathway Analysis in Alzheimer's Disease. So to preface, I want to briefly explain Alzheimer's disease, or commonly called AD. Alzheimer's disease is a disease that impacts memory and behavior in a very progressive manner. So this disease, of course, originates in the brain and actually affects the entire body as it progresses. AD is commonly linked to plaque formation and tangle formation, and is also involved in protein accumulation pathology. So just a brief visual, you can see this picture with the sort of brown spots, which represent the plaques and tangles within a cell and its neurons. And so Alzheimer's disease is actually an age-related disease, which if you really think about it in its context, you think about body systems and how they don't function as well as they once did, which is really interesting because then we are forced to look at different systems and how they age and don't function properly. So the most important mechanism that I evaluated is the lysosome. So the lysosome is a cell organelle that contains degrading enzymes. And so these lysosomes are, of course, within these cells. And then within these lysosomes are enzymes. So a big aspect of my study is this idea of the cathepsin enzyme. And these enzymes have been linked to Alzheimer's disease. And the reason we look at these specific enzymes is because they are responsible for degrading various plaques and tangle formations within Alzheimer's disease. So of course, we know the amyloid beta plaque is a very important aspect of Alzheimer's disease and is of course a precursor for this disease. So it is of course evident in this lysosomal evaluation. And in order to evaluate Alzheimer's disease and the lysosome in correlation to one another, I used bioinformatics. And bioinformatics is often titled an interdisciplinary tool, which combines, most importantly, biology, computer science, and mathematics, but of course it includes very different aspects of science as well. And the pros of using bioinformatics is that it is financially advantageous. So if you were to compare one lab experiment, it is way more costly than it is to use bioinformatics, which is a cumulative and apprehensive study tool. Bioinformatics uses pre-existing and peer-reviewed literature. So the idea of pre-existing and peer-reviewed literature is really important because having already existing data is very helpful if you are approaching an apprehensive idea or question. And it has peer-reviewed literature, which means that scientists in the field have already accepted and supported that such studies within this database tool are, of course, reliable. And bioinformatics also utilizes a very large sample size, which is important because the results of any bioinformatics study can have much more support versus just one lab experiment. And the specific tool I used with Dr. Smith is the QIAGEN IPA, which has, I believe, over 6 million findings. So currently in the field, and of course, starting off very early in the 2000s, Dr. Barr had started work on Alzheimer's disease and the lysosome. So he used a model organism, specifically protein accumulation induced hippocampal slice structures, and he actually applied this PADK, which is a weak inhibitor, to the lysosome in order to modulate it. So this PADK weak inhibitor was actually found to disrupt synaptic integrity. And this plaque specifically is the amyloid beta protein that we all know of, and it's correlated to Alzheimer's disease. And subsequently, when the PADK was inserted into these splice structures, there was a 3 to 4-fold increase of cathepsin and cathepsin B and cathepsin D, which are the modulated enzymes in this experiment. And there's a brief picture on the bottom that represents the correlation between the PADK inhibitor and the cathepsin enzyme. So from there, Dr. Barr also questioned the role of cathepsin B more specifically. And as a follow-up, he continued working on transgenic mouse models as well as hippocampal slice structures once again. And he modulated these model organisms using, once again, PADK, but also E64D, which is another weak inhibitor. And it was found that these inhibitors increased the presence of cathepsin B and therefore decreased the presence of the plaques. So I want to reiterate sort of three key components in these types of studies, the first being inhibitors, the second being enzymes, and the third being these plaques. So when you increase the presence of an inhibitor, you therefore increase the presence of cathepsins, which therefore increases, or rather decreases, the presence of the amyloid beta protein plaques. Other work by Dr. Barr has, of course, related Alzheimer's to the lysosome, including cathepsins and other important molecules that have played a very important role in my study. So this leads me to the gap in knowledge, which is that few known studies have explored linkages among various inhibitory molecules, cathepsin enzymes, beta proteins, and transporter molecules in Alzheimer's disease using bioinformatics. And the purpose of this study is therefore to understand and characterize the connections among various Alzheimer's disease molecules. And my hypothesis is that the genes identified from various peer-reviewed articles are going to be implicated in the lysosomal pathway in Alzheimer's disease. So my role in the study began with extensive peer-reviewed literature search, which took a various number of months. And I used various search engines, including Google Scholar, PubMed, and ScienceDirect. And I specifically typed in keywords such as Alzheimer's and the lysosome in order to find what I was looking for. And using all this research, I formulated a molecules of interest list, which is sort of the driving aspect of this study. And this molecules of interest list is divided into 3 subcategories just for organizational purposes. The first being molecules related to Alzheimer's, the second being molecules related to the lysosome, and the third being molecules related to both Alzheimer's and the lysosome. And once I had this list, I then worked with Dr. Smith on the in silico analysis using QIAGEN Ingenuity Pathway Analysis, which I will refer to as IPA. And from there, I selected what I thought the most important results were, and I independently analyzed these results with, of course, the guidance of Dr. Smith. Very confusing. So the molecules of interest list that I had previously mentioned is seen here. I have a total of 24 molecules, and the colors correspond to the subject, or rather category, in the molecules of interest list. So the actual procedure, which was done in IPA, was mostly twofold. So the molecular pathway analysis started with the inputting of the molecules of interest list. And I, of course, opened the pathway, applied standard filters, and very importantly, added indirect relationships to further contextualize the molecules in relation to one another. And the second aspect of the methods is the core analysis, which of course also started with inputting the molecules of interest list and applied standard filters to produce the results. So first is the molecular pathway analysis, which is really a visual that helps you understand the relationship between all the molecules of interest. And seen in red are the specific cathepsin enzymes that I had decided to put in this list. And you can see that at the top, you can see that the cathepsin enzymes are not only related to each other, but also connect with different lysosomal molecules as well as inhibitory molecules. And once again, if you were to focus on the green circles, which are the inhibitory molecules, you see that they have connections to not only cathepsins, but also the lysosomal molecules. And lastly, the circled in blue are the lysosomal molecules, which have implications with not only inhibitors, but also cathepsins. And the second aspect is the core analysis, which is presented in a histogram. And furthest to the left is actually the highest p-value, so the most statistically significant pathways. And so these pathways are called canonical pathways. And just a brief sort of definition of that, it's pre-existing pathways with known molecules, and they can range anywhere from a dozen molecules to hundreds of molecules. So the two that I have selected to focus on are the phagosome maturation pathway and amyloid processing pathways. And just to further explain or give you more context of these pathways, this is the phagosome maturation pathway, which has, I'm sure, dozens of molecules. But if you look more closely, in purple is LAMP1 and cathepsin, which are molecules within the data set. And therefore, they have lysosomal implications and degrading qualities, meaning that we can look at the phagosome maturation pathway for more exploration involved in Alzheimer's. And similarly, the amyloid processing pathway has dozens of molecules, but if you look closely, gamma secretase and beta secretase are within my data set, and therefore they have lysosomal implications and degrading qualities. So therefore, we can also look at the amyloid processing pathway as a future mechanism for Alzheimer's. So therefore, my hypothesis that the genes identified from various peer-reviewed articles are implicated in lysosomal pathway in Alzheimer's disease has been supported. And the findings of this study align with the results of the work of Dr. Barr, as well as other scientists in the field, in that the cathepsins are implicated in the lysosome and various degrading pathways. So it is important to understand that a bioinformatics study is, of course, limited by the studies that it is supported by. So the specific IPA has over 6 million findings. So any limitations from those experiments will also therefore apply to this bioinformatics study. Additionally, the data set was rather small, using only 24 molecules, which of course could be expanded in the future. And also the publications were limited from 2000 to the present, so anything before that was not included. And finally, the p-value was less than 0.05 for all results. So everything was in fact statistically significant. And like I said, in the future, I can expand the molecules of interest list, investigate new pathways, and potentially enter a lab to get hands-on with these mechanisms. So overall, the lysosome has been further supported in Alzheimer's disease. Potential inhibitors have been discovered in order to combat protein accumulation for drug production within Alzheimer's, as well as specific pathways have been pointed to for further exploration for Alzheimer's therapies. So I'd like to thank my mentors, Dr. Barr and Dr. Smith, both who are on the call today. Thank you again. And I want to thank my teacher from my school, as well as my family and friends for constantly supporting me. And here are my list of crazy references. And I want to thank you all for listening to my project, and I will take time for questions if there are any.
And I guess maybe before we start, I just, again, want to reiterate and thank our students for joining us today and for sharing their work, and also for being so interested and committed and engaged with dementia research. I think it's really remarkable and gives us a lot of hope for making an impact on this disease. So thank you all.
And Kyle, I'd like to add my thanks to the students as well. This is really amazing and impressive. Y'all have done so well and presented so clearly. I'm thinking back to my high school days, and I'm in total awe of your capabilities. I don't think I could have done that sort of work at your stage, so I'm very impressed. So Danny, when you were thinking about expanding the list, how would you go about that? And do you have ideas about what types of molecules that you would add to that list?
So I think my list, one area that was sort of not lacking, but something that I want to specifically expand upon is are sort of just these molecules that are solely related to Alzheimer's. So the reason I think that this would be interesting is because it would further support different Alzheimer's molecules and sort of question it in this context of lysosomal pathways or degrading pathways. And of course, this would have to be driven by a lot of continuous research through different articles online.
So I had a question for Bianca. Just between, you've done this in sort of different contexts. So some of it was, providing activities that individuals can, take home with them, right? If they're living with their caregiver, living at home. And then some of the activities you've been doing have been sort of in a, institutional care facility, right? And so I was just curious about your perceptions or what you've learned about maybe some of the differences in terms of providing activities and items in sort of for individuals who are living at home versus in a care facility, and how that sort of impacts how you think about this and how you think about the interventions.
Yeah, so when I visited a lot of these facilities, my eyes were definitely more open to like geriatric care, like specifically. So I remember going to like the Grace Richard Timon community, and I remember seeing a lot of patients, and they told me, like it's really lonely in these communities. Social isolation is a big factor that can contribute to Alzheimer's disease, can lead to the disease to progress faster. So as I've talked with these patients in these facilities in like a group setting, I've just found that, like connections within these facilities, like having like a support network in these facilities, it's like super, super important. So by, engaging with everyone in a group format with like poetry reading or like singing, I think that's really been helpful for them to like talk and engage with other people. But on the other hand, when I'm providing, these activities like for these individuals who live at home, I found that they can be more effective because it's more personalized that way. You're not working with an entire group, and everyone has different kind of interest, skills, like cognitive stages. So I feel like there's like downsides and there's also positives to each setting. That's why I like do both.
I guess for Danny, how has this been working remotely on this project? I know like everybody's gotten better at remote work, but it's it's sort of a different world. And so, how's that been for you, and maybe how's that been, Sylvia, on your end as well?
On my end, I mean, I'm very, very appreciative of both of my mentors. And I think my specific methodology made it really possible to do it online, given that bioinformatics is a computational tool. So the outlet we're on now, Zoom, is actually what Dr. Smith and I used for to conduct this study specifically. So everything went pretty smoothly. Yeah.
Yes, actually, I only had access to one license for this software. It's very, very, very expensive. I think it's $20,000 per person per year. And so even if she had been here, we would have had to do it together. So we did it on Zoom, and it worked out all right.
And
Well,
there's a question in the chat. I'm sorry, Cal, didn't mean to interrupt.
Oh, no, I was just saying, I think that the sort of, Zoomification of meetings and research, and, even our lab meetings every other week are on Zoom, have kind of opened up more opportunities for young investigators to get involved, particularly, for those who either don't live near a major academic center, or maybe who do, but, want to work with a specific person or in a specific area of focus that they're passionate about. So I think that's good. So let's see. Oh yeah, okay. Question in the chat for Kajal. Let's see. Can you expand a bit on microglia therapeutics? Go for it.
So this is just based on what I know so far, but Dr. Walsh, please feel free to add on. There's lots of drugs targeting something when it comes to Alzheimer's disease, but we've looked at if it's done in reverse, it could actually help glioblastoma too, and the same the other way. And also many of the challenges that GBM researchers are facing are things that AD researchers are actually doing. So these targets are potentially shared, but that's pretty surface level. So Dr. Walsh, if you want to add on to that.
Yeah, I mean, I think the idea is, if you're trying to drive something one direction and you're an Alzheimer's researcher, and there's a bunch of glioblastoma researchers that you don't interact with, who are targeting the same molecules and the same cell types, but they're trying to drive them the opposite direction, there's probably a lot of synergy that can be gained there that's sort of going untapped, right? And so, even in our experience here, like constructing antibodies to things that are sort of to drive microglia to be more inflammatory, but to do a lot of that type of work, you have to overcome their sort of anti-inflammatory nature that I think is potentially a little more tractable in the Alzheimer's space. So I don't think, based on the sort of opposite directionality, I doubt that, you could have a drug that worked for one and the other, but you might have a failed drug for one, or a drug that, is known to exacerbate one that could potentially have some impact on the other. Okay, we're at 5, Jan. So if anybody else has any questions, speak now, forever hold your peace. Otherwise, I think, again, we just want to thank the group and thank these great young investigators, and wish them all of the luck, both with their current projects as well as their college applications and in some of their cases, and in all their future endeavors. So,
really great job, everyone. Very impressive. Well done.
Thank you.